Hello dear students, welcome to my tutorials. This would be one of the tutorials that will give you the complete solution to your NCERT. So please do subscribe it immediately. In today's video, we will study about RNA, what is it and what are the various types of it. So let's get started. First, we will see what is the full form of RNA. So RNA stands for ribonucleic acid. If you have seen all my previous videos, it would be quite easy for you to understand. Otherwise, if you are straightforward coming to this video, please do watch out the one in which I have described the structure of DNA, and that is the very first video of this chapter. Now the other thing about RNA is, till now you can say that it is single-stranded. Means DNA was double helical, but RNA is single-stranded. It is also a polynucleotidic structure, means this is also a polymer. And you know that polymers are the long molecules. They are formed by the aggregation of monomers, right? So monomer in the case of RNA also, it is nucleotide. And a nucleotide is a one that is formed by the combination of base, sugar and phosphate. Okay, this we have already studied in DNA also. Here also these three components will be there. Now if we come to bases, again two types of bases will be there in RNA. One will be known as purines and another will be known as pyrimidines. Okay, purines, again these are the double ringed heterocyclic structures, whereas pyrimidines are the heterocyclic ring structures, but the ring will only be one in them, right? So purines include adenine and guanine. Adenine will be abbreviated as A, guanine will be abbreviated as G. When it comes to pyrimidine, RNA has cytosine abbreviated as C and uracil abbreviated as U. So here if you could notice, this nitrogenous base is different from DNA. There in DNA, it was thymine, right? It was thymine in place of uracil, but in RNA, uracil is there. This difference is very important. So whenever you will be making the bases from template strand of DNA and you are about to make messenger RNA, make sure you remember this thing, and you are never ever supposed to write T in any of the RNA sequences. Now let's come on to the sugar. Sugar in the case of RNA, it is ribose. This has been told in DNA also. Let me tell you a short step of it as well. So this is our pentose sugar, and pentose sugar is a one that has 5 carbons into it. Now ribose at its C2 position, it contains hydroxyl group, whereas deoxyribose sugar of DNA, it only contains the H group at C2 position. Now this C2 with OH, this is present in the case of ribose sugar, and ribose is an example of pentose sugar. Now the presence of this OH group at C2, it makes the ribose quite unstable. And you know, over a period of time when Earth evolved, the first genetic material was RNA. That is why we can say that RNA is more primitive, but because of its C2 position, it was unstable. So over a period of time, which became DNA material? Yes, DNA became the genetic material. Phosphate group will be same. Okay, so nucleotide will be formed first, then many nucleotides will be combining and the polynucleotidic structure will be formed. So suppose this is the sugar, right? I have already told the positions C1, C2, C3, C4, and this will be the C5. Okay, so if you are to make the nucleotide, phosphate group will be attaching here, and at C1 position base will be there. Okay, so the similar type of attachment will go on further. So if you want to attach one more nucleotide to this nucleotide, remember at C3 position you will attach the phosphate, and then you will make like this. So this way, if you go on making it, you will get a polynucleotidic structure. Remember, this is our C5 position, this is our C3 position. So if you want to make continuously, you can make it. Okay, so this way again, if you want to make, you will make phosphate group here, and so on and so forth you will proceed, right? Now another thing, in DNA we talked about purines are always equal to pyrimidines, but there is no case as such in RNA, right? So because RNA is single stranded, and Chargaff's rule which stated that the amount of purines and pyrimidines, they should be equal, that Chargaff's rule will not be followed on RNA because it is single stranded, whereas it was followed in DNA because it was double stranded. So this was all regarding the RNA, its structure, its instability. Now we will study the major types of RNA. So our next heading is types of RNA. More differences between DNA and RNA will be recorded separate because this is again an important topic. So there I will speak up in detail about it. Do check it out. So types of RNA includes ribosomal RNA, and here R has to be written small and it is indicating ribosomal. The second is messenger RNA. Here M means messenger. We will be discussing each one by one. tRNA. T stands for transfer RNA. snRNA means small nuclear RNA. Remember, whenever you are writing the types of RNA, do write the type in small letters. See here, R, M, T, SN. This type has been written in small, but RNA has to be written in capitals always. So you can notice in all these four types, I have written RNA as the capital one. In this chapter, we are to read about these four. However, there are other types of RNAs are also there, but we will be discussing these four in relation to molecular basis of inheritance. So let's get started with our first RNA, that is ribosomal RNA. Now ribosomes, as you know, they are the protein factories, and the ribosomal RNA would be the one that will be associated with the ribosomes. Now almost about 70 to 80% RNA in a cell is ribosomal. So if a question comes in the board examination among all the types of RNA, which one is in maximum amount in a cell? So answer is ribosomal RNA. Okay, so ribosomal RNA, it is associated with the ribosome. Ribosomes are the protein factories of the cell. Now you must be knowing, because this topic was there in the grade 11, in prokaryotes and in eukaryotes, ribosomes are different. In prokaryotes, they are 70S. In eukaryotes, they are 80S. If we bifurcate their larger and smaller unit, then it is 50S in the case of prokaryotes, the larger subunit, and the smaller subunit is of 30S. Okay, so a functional ribosome is a one in which larger subunit and smaller subunit, they are attached to each other. Otherwise, whenever they are separated, they are not functional. Whereas in the 80S, the larger subunit is of 60S and the smaller subunit is of 40S. Okay, so S here, the S unit is there after 80. It means sieved Berg unit. This is also known as sedimentation coefficient. Okay, sedimentation coefficient means this will tell us that what is the sedimentation rate of this ribosome. Accordingly, they are given the values, right? So here you can write larger subunit and here it will be our smaller subunit. Now ribosomal RNA, if we first talk about eukaryotes, then it could be 28S, 5.8S or 5S, and it could be 18S2. Okay, so ribosomal RNA, it could be of these four types. The ones which are written here, they are all associated with the larger subunit. See carefully, I have told that we are talking about ribosomal RNA in the case of eukaryotes, right? So these units, these ribosomal RNA, they are associated with the large unit, whereas the 18S1, which has been written separately here, this will be associated with the smaller subunit. Okay, this we have to learn. This is important. Now let's come on to the prokaryotic ribosomal RNA. So here we will be talking about 23S, we will be talking about 5S, and we will be talking about 16S. So in prokaryotes, ribosomal RNA could be 23S, 5S, or it could be 16S, right? So these two which are written together, they are associated with the larger subunit, whereas the 16S is associated with the smaller subunit. This you need to learn. Okay, so we are done that ribosomal RNA could be of different types depending upon the larger subunit and the smaller subunit they are associated. Let's move on to another type of RNA, and this is messenger RNA. We do know a bit about it because we are already done with transcription. In that we have read about it, and it is written as mRNA in the abbreviated form. Now why are we calling it as messenger? Because it is bringing the information from the DNA and it is passing the same information in the cytoplasm. So right, it is working as a messenger. It is bringing the information from somewhere and it is acting somewhere. That's why it is known as messenger of the cell. So messenger RNA constitutes about only 5% of RNA in a cell. Messenger RNA is a one which is formed by the process of transcription, right? And it undergoes processing and it becomes functional. Now once RNA is formed from the DNA, it may process in various ways to form the ribosomal RNA, to form the messenger RNA, or to form the tRNA. Okay. Now this RNA, it could be polycistronic. Polycistronic means if it would be synthesizing, one messenger RNA would be synthesizing many polypeptides, right? So as you will see in the Lac-Operon theory, that from one messenger RNA, 3 polypeptides will be synthesized. So this type is present in the eukaryotes. Again, I say the difference between the polycistronic and monocistronic, they are important from board perspective. The another type of messenger RNA is monocistronic, and monocistronic means only one polypeptide will be synthesized, and this is the case in eukaryotes. So do remember, monocistronic messenger RNA is the characteristic feature of eukaryotes, whereas polycistronic is the characteristic of the prokaryotes. Now we have seen that heterogeneous RNA, it changes to messenger RNA by the process of splicing, capping and tailing, right? This we have seen in the second video of transcription. If you haven't watched that, please do watch it. It will help you in a better understanding. Now, heterogeneous RNA changes to messenger RNA because there is a need, because heterogeneous RNA, if it will come out of the nucleus in the cytoplasm, it is going to get churned, it is going to get digested by the cytoplasmic enzymes. Just to avoid this, it has to be processed. Now, when processing is happening, it will happen by 3 steps: splicing, capping and tailing. Mind you, these steps will occur in the eukaryotes. Prokaryotes don't go for such processing. They immediately transcribe, and after transcription they immediately go for translation. Means in between there is no processing present. Okay. Now another thing which is related to messenger RNA is the UTR. In board, they may give you a messenger RNA sequence and they may ask you to find out the UTR region in the given messenger RNA. Okay, so what is this UTR? UTR stands for untranslated region. Okay, so this means this region would be the one that is not going to do translation, means it will not help in the protein synthesis. So let me make a sequence for you and we will find out the UTR in that sequence. So this is messenger RNA, which is 5 to 3 prime, and let me write the bases in it. Suppose I am writing randomly bases. Okay, and here I end up. Okay, now your first job is to find the initiator codon. Okay, initiator codon. So if you have not seen my video on the genetic code, do watch it out. There I have talked about what is initiator codon. Here to let you know, initiator codon would be the one that will start the process of translation, because translation is going to happen in 3 steps: initiation, elongation and termination. So someone has to initiate. Initiator codon will do the initiation. So here we can quickly guess that this is AUG, and this always acts as our initiator codon. Means the process of translation will start from AUG. Another thing that we need to find out is our terminating codon. Okay, terminating codon. So these are the tricks to find out. Okay, first I have told you, you need to find the initiator codon. Second job is to find the terminating codon, and the terminating codon here is UAG. Okay, so this would be the one that will terminate the process. Okay, because it is an example of terminating codon. Let me add few more base here. Now you have seen that this portion would be the one that will undergo the translation process. Okay, but here the regions which are happening just prior to it, which are colored green, these would be the ones that will not undergo translation. So there are two UTRs in the given sequence. This is also your UTR, untranslated region, because there is no initiator codon seen for these, and this is also the UTR. This is again very important. As I said few minutes ago, please do remember, it's very easy. Take the notebook, write the sequence, find the initiator codon, find the terminating codon. This would be acting as that segment that will help in the translation. The regions occurring prior to the initiator codon and after the terminating codon, they would be the one that will be counted as UTRs. Okay, now we are almost done with messenger RNA. In this video, I will not talk about snRNA because I have already talked about it in the splicing mechanism that was recorded in transcription video number 2. So please do check out its role there. The last type of RNA which we need to discuss right now is transfer RNA. Transfer RNA is also important for the examination, for the competition, because questions frequently asked on this. Transfer RNA is abbreviated as tRNA. It is also known as soluble RNA, right? It is also known as adapter molecule. These are the various names of transfer RNA, okay? So if you get a question in exam, draw the adapter molecule. Please quickly recollect that they are asking about transfer RNA. They can also ask you a question as which form of RNA is soluble in nature. So you will write the answer as transfer RNA. Now transfer RNA, it is made up of 70 to 80 nucleotides. RNA, you know, it is single-stranded RNA, but here the peculiarity in tRNA is that the molecule gets folded in such a peculiar way that it looks as if it is double-stranded. Because the bases, while folding, they are complementary, so they form hydrogen bond between them. Now the structure is also important, and there are various arms in it which are also important. Remember, whenever you are going to start the transfer RNA, you will make from the 5' end. So make sure this is your 5' end, and always you have to show the nucleotide G or guanine at the 5' end. Okay, do follow the steps, and by this you will get the full marks. Okay, these are the various arms which we are required to make. And here there would be one more thing. There will be a variable arm, and then it will end up here, and this will be our end, that is going to end with a sequence of CCA, and OH end will be there, and this is our 3' end. Okay, so we do have how many arms? One arm is this, okay, and this will also be counted as one of the important end of tRNA. Okay, let's discuss one by one. So this is our anticodon arm. Okay, this is our anticodon arm, and this will be the one that will be binding to the messenger RNA. So if I draw messenger RNA here, and suppose messenger RNA has a codon on it, AUG, then this of course has codon sequences. So here we would be making the complementary bases will be there. So here for A it will be U, for U it will be A, for G it will be C. So this UAC is anticodon. So anticodons will be found in the anticodon arm, and this arm will be the one that will be binding to the messenger RNA during the process of translation. Okay. Now this arm here is a variable arm, means the length of it varies. Okay, here it is, this one could be your second, okay, which you have to draw. This could be your third, and this is known as DHU. Okay, it means dihydrouridine. Certain bases get modified in the transfer RNA, and that's what DHU is. Okay, so DHU is actually forming a loop like structure, right? So sometimes we call it as DHU loop also. This is our T psi C. Okay, T psi C, and this would be the one that will be binding to the ribosomes during the process of translation, whereas DHU loop, it has the function of binding to the amino acids activating enzymes. For now, you can just learn that this will bind to amino acid. Here AA means amino acid activating enzymes. Details of it will be discussed in the translation process. This is the one process where the amino acids will be activated. So opposite to the anticodon loop, amino acids are activated, right? So amino acids activated here. Okay, this is the structure of tRNA, which is showing DHU loop, anticodon loop with its arm, right? T psi C loop, variable arm, because the number of bases in it, they may vary, and the above arm, that means the one just opposite to the anticodon loop or arm, this is known as the place where the amino acids will be activated for the translation process. Okay, now here in between it is seen that bases are as per the complementary base pairing rule. That is why here these lines are being drawn, which are showing hydrogen bond. So this structure of tRNA is also important for board. Question may come for 2 marks and they may ask you the structure of tRNA. Hope this was helpful. Stay tuned for more videos. Thank you.